Hello, hello, hello.
Hello.
So, welcome to today's class. In the last class, we had worked with the data frames and lists in R, and I also posted some. So, I've also posted some work, R codes and an exercise to uh, exercise using which you can practice. So, in today's class, we'll be going back to a bit of a theory. We'll be looking into temperature response functions. So, temperature is a very important parameter when it comes to plant physiological processes, because temperature affects almost all of the biochemical reactions happening in plants. So, models use these temperature response functions to predict physiological processes like photosynthesis and respiration. So, here we'll be covering some key temperature response equations that are generally used when it comes to temperature response functions. Um, Hi, Ananta. Today we'll be covering temperature response functions. So, the first function that we're going to look into is the Arrhenius equation. So, this equation was developed by this person, Svante Arrhenius, based on the work of chemist Jacobus Henricus van 't Hoff. This equation is important because this equation is demonstrating the temperature dependence of reaction, basically as enzymatic reactions like photosynthesis and respiration. Photosynthesis is catalyzed by the Rubisco enzyme, and this is highly dependent on temperature. And this Arrhenius equation is helping us to relate this equation, the enzymatic reactions, to temperature. Arrhenius equation is given by this formula here. Mm, K is equal to A e to the power minus EA by RT. Now, K is the rate constant, A is the frequency factor, EA is the activation energy, R is the ideal gas constant, and T is the absolute temperature in Kelvin. Now, when we talk about T over here, we are talking about the temperature of the leaf because we are dealing with leaf response, the photosynthetic response in leaves. Now, if you look at this equation, if this value of t keeps on increasing, at say as t approaches infinity, this entire value becomes zero because anything divided by infinity is zero. So e to the power minus zero will be equal to 1. So k a is basically a can be expressed as the rate constant when there is no limit to the increase in temperature. And A is not a value that is very easy to obtain because again we say it is based on a lot of molecular molecular collision and stuff like that. So it's not a very easy parameter to obtain. So what we do is, in particularly in the model BioPro, what we do is we are redefining it as a constant called known constant called K naught at a reference temperature T ref, generally assumed at 25 degrees Celsius. So here what we do is at reference temperature T ref, the rate constant we define it as K naught. So K naught will be equal to A into e to the power minus EA by R, except that the reference temperature considered here is T ref. Now, if you bring this term to K naught over here, if you bring this term to this side, it becomes A is equal to K naught into e to the power EA by RT ref, because this minus this will become equal to K naught by uh, e to the power minus K by RT. And when this thing comes to the numerator, it will become K naught into e to the power K by RT. So here we can write A is equal to K naught into e to the power EA by R into T reference. Now when we substitute this A value in this first equation over here, K value, we will get K is equal to K naught into e to the power EA by R T rep into e to the power minus EA by RT. Now, when we rearrange all these terms here, we get that K naught into EA by RTF minus EA by RT is the final equation we are getting. So instead of dealing with this EA by R at T reference temperature, we are always going to keep the reference temperature say as 25 degrees. So we can obtain this value by knowing the reference temperature, and for that we will keep this value as a parameter, dimensionless parameter C, and then the equation can be written as K is equal to K naught into e to the power C minus EA by R. So this is the Arrhenius equation, and this is the Arrhenius equation that is modified so that we don't have to deal with the frequency factor in the Arrhenius equation by doing some assumption with respect to using a reference temperature. Now, in our C3 photosynthesis, where is this Arrhenius equation used? It is used when we estimate the Rubisco carboxylation. We learned in the FPCB model that the Rubisco carboxylation is given by this equation, and here there are a lot of parameters like VC max, tau star, or carbon dioxide condensation point, the Michaelis-Menten constant for carbon dioxide as well as oxygen. So this term you see Kc over here is a function of temperature, and it is represented by this Arrhenius equation, where here you can see this is an example for this equation where the parameter C is equal to 38.05. The parameter EA is equal to 79.43. If you see the Arrhenius equation on the right, here C is equal to 38.05 and EA is equal to 79.43. R is the ideal gas constant and TL is the temperature at which we require to find the Kc parameter. Similarly, K naught is also represented, but you can see that the C value is changing over here. And similarly, EA is also changing. So EA is the activation energy and C is the dimensionless parameter that we have found out to so that we don't have to deal with the frequency factor in Arrhenius equation. Similarly, tau star also, carbon dioxide compensation point also can be found out using the Arrhenius equation as a function of temperature. Now here if you see this K naught term is all equal to 1. So in these equations, at the reference temperature, that is 25 degrees Celsius, Kc value is K naught and tau star value will be equal to 1 is what is assumed over here. Now at the same time, if you look at the Vc max term is equal to some Vc max multiplied by this Arrhenius function. This Vc max is a maximum Rubisco carboxylation rate that is measured at a particular temperature. Now with that value, we can find out what is the Vc max value for any temperature that we require by using this equation. Now again, in when it comes to the RUBP regenerated phase, we have the term J, we have the term carbon dioxide compensation point, and in order to find J, we need the term J max, that is the maximum electron transport. This is the rate of electron transport. but maximum rate of electron transport is given by Jmax. We also have a curvature term called theta, and we also have a term called, yeah, we'll come to that. We have, okay, so we have this Jmax term, which is again expressed as a function of the Arrhenius equation. Here you can see Jmax is the maximum rate of electron transport at a reference temperature, say 25 degrees Celsius. And we can find out this value, the Jmax value at any temperature by putting it in this equation. So here the activation energy is equal to 43.54 and 17.57 is equal to C value. Similarly, RD also can be found like that. So if you look at it, all these values, the dark respiration, the RUBP regenerative limited photosynthetic rate, the Rubisco carboxylation rate, all of these are dependent on temperature because it is all enzymatic driven reactions. So this is how we relate the enzymatic reactions in photosynthesis, particularly Rubisco and RUBP regenerated phase equations with temperature using the Arrhenius equation. Now, in the same way, similar to Arrhenius equation, there is also another equation that is used called the Johnson-Eyring-Williams model. In here, whatever is a parameter is expressed as a function of temperature. So this can be any function. Say if it is Vc max, Vc max as a function of a temperature will be equal to the absolute temperature in Kelvin into e to the power C minus HA by RT. Again, C is the dimensionless empirical parameter that we are using to adjust for the reference temperature. HA is the activation energy, RT is the ideal gas constant into the temperature in kelvins. And here we also have a numerator where we are dividing the term by 1 plus e to the power S by R, where R is the ideal gas constant and S is the entropy that is affecting the deactivation term. So similar to having an activation energy in the numerator, in this equation, John-Eyring-Williams model, we also have a deactivation energy that is utilized to find the value of a parameter with respect to as a parameter dependent on temperature. Now, in photosynthesis, particularly in C3 photosynthesis, we use this equation. We are using this equation in finding the TPU rate. So if you see here, similar to how we found out the Vc max and Jmax value, we'll find out the maximum rate of triose phosphate utilization by multiplying it with the value in the reference temperature with the temperature of the leaf into e to the power 25.5 will be the C value, 62.99 will be the activation energy, R and TL we know, and here this value S by R. Now why TL is coming in the numerator is because this whole term is taken, this entire term is taken as one term. This entire term taken as one term, so it will be S into T minus HD by R into T. So here this S term is 0.588 and TL we know HD is equal to 182.14 and RT is again ideal gas constant and the leaf temperature in Kelvin. All of these the temperature has to be substituted in terms of Kelvin. Now, another temperature response function that is utilized in C3 photosynthesis is the polynomial temperature response function. So, polynomial temperature response function is nothing but the temperature response is expressed in terms of a polynomial equation. We know polynomial equation in the form of A plus Bx plus Cx square. And here again, all these A, B, and C, which is represented by C0, C1, and C2. C naught is the baseline rate, C1 is the first order rate, and C2 will be the second order coefficient. And the first order coefficient, which is C1, it is depicting how this function will change linearly with respect to the temperature. And so if C1 is positive, the response will keep increasing. The function, this function particularly, will keep on increasing. If it is negative, it means it will keep on decreasing with temperature. Now, in the same way, this C2 function is the second order coefficient where it is accounting for the non-linear temperature dependence. Now, you can see this function f of t will vary in a non-linear fashion. If you are looking at this term here, it is varying linearly with temperature, but here it is varying non-linearly with temperature. So if C2 is negative, it means the response follow a parabolic shape, that is peaking at an optimal temperature and then declining. It will go like this. This one will go like this. And then if it is positive, you will see that the function curves upwards, meaning the process is accelerating at higher temperature. Sorry, if it is positive, it will go like this. If it is negative, it will go like this. So um, So unlike Arrhenius and Johnson-Eyring-Williams models, which are both enzyme kinetic based models, polynomial functions are offering a simpler empirical fit to temperature data. So this function can be represented in terms of a temperature or as a function of temperature using this first order, which is the linear thing, and also the second order coefficient where it is considering the variation of the function with respect to temperature in a non-linear manner. So if this will be how the curve will be if it is positive, the first order coefficient, and how it will be negative in the second if the first order coefficient is negative, and if C2 is positive, that is the second order coefficient is positive, it will become like this. And if C2 is negative, it will be a downward curve. Now, in photosynthesis, particularly in all those photosynthetic terms that we talked about, we use this function majorly in the RUBP regeneration phase. So earlier, I had shown that Jmax value is Jmax value is an Arrhenius varying with the temperature and expressed as a function of temperature using Arrhenius equation. But then again, there are two more terms, the theta psi and the phi psi values. These two values are expressed as as a function of temperature using the polynomial temperature response function. So here theta, you can see this is the baseline theta. This is the first order coefficient that is relating this function theta psi linearly with the temperature, and this is the second order coefficient that is representing theta psi as a function of temperature, which is varying non-linearly with temperature. And again, Q2 is a function of phi ps2, and phi ps2 is also expressed using this polynomial temperature response function. This will be the baseline value. This will be the first order coefficient, and this will be the second order coefficient. So what you see here is both of these equations have negative uh, coefficient, negative second order coefficient, and you can see negative second order coefficient has this kind of a line. So in this case, whenever the temperature is increasing, the tendency of this value is to follow this kind of a path with temperature. Now, how do we obtain all these parameters like the dimensionless parameters, the activation energy, C naught, C1, etc. So we can do this by measuring the physiological responses. I had explained about the ACI EQ curve. So we can do this by finding out ACI curves at different temperature. Now for usually when we measure ACI curves, we are going to measuring at a particular temperature, particular light, particular humidity, and we will only keep changing the CO2 values. Uh, this when multiple ACI curves are taken at different temperatures, say T1, T2, T3, then we can fit these measured curves to all these ACI curves to obtain how is the C value, EA value all varying with temperature. Now another, we are going to fit the FPCB model with the measured curves to find this, find these coefficients that is relating a parameter with temperature. Now, if you had seen earlier, all of these parameters are varying. It is not the same for everything. Even if you look at these terms over here, all of the S value is varying, the C value is varying, the EA value is varying. If you look here also, for in one equation itself, all these values are varying uh, with respect to temperature. So this is a highly variable highly variable constant. And in order to obtain this, what we need to do is one way is to generate a lot of ACI curves at different temperature. Like you start with a 20 degree, then you measure four ACI curves at 20 degrees. Then you measure four ACI curves at 25 degrees. Then you change it to 30 degrees, then 35. So multiple curves at different temperature levels will help you obtain using multiple curves that is obtained at different temperature, that way ACI curve, you can obtain the CEA and all the temperature response parameters. Now another way is to uh, generate the temperature response curve. So we had learned about ACI curves, which are curves where net assimilation is varying with intercellular carbon dioxide concentration. We had also learned about AQ curves, where the net assimilation is varying as a function of light. Now AT curves is the net photosynthetic rate or assimilation rate varying with temperature. So similar to how we measured ACI curves at different carbon dioxide concentration, we can also measure AT curves. That is, we are going to increase the temperature alone from, you can see in this one, the values are varying from 5 as low as 5 to 45. So the temperature can be measured from 5 to 45 by changing the temperature from 5 to 45, keeping the carbon dioxide level at 300 or 150 and light at say 1800 and RH at some value. You can change only the values of temperature and measure the photosynthetic rate. And these measurements can be made, I mean, multiple curves can be made, and you can measure it for different levels of carbon dioxide to analyze how these from such AT curves, we can obtain the um temperature response parameters. Okay, so now I'll take you to the Biocro model to see how we can, how the model and the modules in it represent this temperature response function. So in Biocro, this temperature response function plays a huge role when it comes to the C3 assimilation module. Okay, so I will start a new R script. I have to check where I'm working, get working directory, and I want to set the working directory to my required, which is this folder. So, and I want to show how the BioCro is making use of this temperature function. So another thing to take note here is BioCro has updated its version. So you, those of you who have already installed it can reinstall this package so that because in the older version, which was 3.1 or something, the temperature response functions were not explicitly available. It was hard coded. You wouldn't have, you wouldn't have the opportunity to change the temperature response parameters like C and activation energy for each for VC max and Jmax. But the updated version has improved that with the possibility to input the C values and EA values corresponding to each of the term. So you will have to reinstall the BioCro package, and once you do it, you will have the updated version available. So first I load the library BioPro, and I want to look into C3 assimilation module. Since we are our focus has altogether been on C3 photosynthesis, I want to get information regarding the C3 assimilation module. So I use this module info function that is available in BioPro to read this C3 assimilation module. And when I run the line, you see that here C3 assimilation module, the input quantities, initially we only had all these input quantities, but now it has an updated to get the G star EA, J max C, J max EA, KC K naught C, K naught EA, theta PS2. Oh, okay, okay. I'm sorry. I think I didn't share the screen. Thank you for letting me know. Is it uh, is it visible now? Okay. If you have it installed, you can just try doing it along with me. Do you have access to a laptop, Ananta, or are you using a phone? Okay, great. So, do you want to try reinstalling BioCro? I mean, do you remember how it is done? If you want to install BioCro again, just go to Tools, Install Packages, and just type in BioCro over here. It'll come. And then install. Because the older version has got a older uh, module, it's good to reinstall the BioPro package. It's a quick process, so you can install it and then load the library in this way. And if there are any errors, please let me know. You can drop in the chat box and I can rectify it. So first we'll do module info, and we are going to type in BioCro semicolon colon and C3 underscore assimilation. So before this, in case you don't know what the names of the modules are in BioPro, you can do it by get all modules um, BioPro. And you see that here you get all of the modules that are available in BioPro. So if you remember about BioPro, I had said that each of the physiological processes in BioPro is represented by a particular module. So if you take out one module alone, you can work with one particular process alone. So here we are working with C3 photosynthesis. So that is why I want to use the C3 assimilation module. And if you copy this whole thing along with the double quotes, and paste it in this module info and run this line, you can get all the details that are related to the module input quantities. Now I had explained um, how we had created data frames and lists and stuffs in R. So now I don't want to keep going to the console to check what are the inputs and what are the outputs. So what I do is I name a variable uh, C3SM module and I save it in that. So here in the data, I get C3SM module. Now, if you see this, if you want to know the data type of this, you just type of C3SM and you see it as a list. So in the list itself, if you want to access the inputs alone, you know what the module name is. You don't want to output alone. You just want to get the inputs. Then I will name another variable inputs C3SM C3_SM module, and then I'll take inputs alone. And then when I run this line, I'll get an input of the input files alone. Now I want it in the form of a data frame, so I will use data.frame C3 assimilation module, and this will come in the form of a data frame. You see, I get all the form in the form of a column. I get all these values in the form of a column. Now, suppose I want to save this file as a Excel file, and I can do that by using this command write.csv. I think I didn't explain. I just told you how to read CSV files. I didn't explain how to write CSV files. So same like reading, you will do write CSV file. Which is the data type you want to save as a CSV file? It is this input C3 assimilation, and then comma. What is the name of the file? I'll put a dot and a slash and write inputs of C3 SM module with the extension .csv, and when I enter, you can see that the folder where I am working in, ISSTA Biocro codes, I would have got the inputs of C3 assimilation. So if I open this, I'm seeing I have all these input. So in later on, even when you're not using BioPro and you want to do some manual calculations or you want to, make use of this information, you can just access this file because I've already saved this file as a CSV file. Okay, now I've got all the inputs. I know the details of this BioPro 3 assimilation. Now, what I want to do is I want to run the C3 assimilation module with some values of uh, all these temperature response and everything. So I had told how we run the module. So in order to run the module, we use this function called module response curve or evaluate module. So I do evaluate module, and the module that I want to evaluate is Biocro 3 assimilation. Now, if you don't know what arguments you need to pass, you can just do this and evaluate module. And they will show you that inside evaluate module, you have to provide the name of the module and the input quantities. Now, there are a lot of input quantities. So what I'm going to do is, BioPro has already been developed for several plants. One such plant is the soybean plant. So all of the parameters that has been used to run the soybean model is available in BioCro itself. So in order to access that, what I'll do is I'll type soybean. So you can see when you type in soybean itself, you see that soybean BioCro model definition, the initial values, parameters, direct modules, differential modules, and a differential equation solver that can be used to run soybean growth simulation using the data from Illinois is stored in this BioCro module. So I can access the parameters of soybean. I can access the weather data that is being used in the simulation of soybean and everything. So what I'll do is soybean parameters and dollar. you can see that the soybean has got the direct modules that are used in the simulation, differential modules, the solver, initial values, and the parameters. So I am interested in the parameters. So I run this, and you can see that the whatever parameters that were used for soybean will be available over here. Now, if you want to again get a more clear view of this, I can again store this in a soybean parameters variable as a data frame, data dot frame as soybean dot parameters. And then you can see I got the soybean parameters. Fortunately, this is in the form of several columns because each parameter is represented as a column header, and we are getting something of this sort. Now, instead of inputting every value within the soybean, what I'm going to do is I'm going to evaluate this module using the soybean parameters. I just want to take in the soybean parameters. So I will evaluate this module with soybean parameters. So in evaluate module, the two arguments required is the module name and the input quantities. So here I will use this evaluate module function that is available in Biochrome. I will put in the name of the module, that is by whenever you put in the name, you can't just put in the name alone. You have to put this Biocro colon C3 underscore assimilation and the input quantity soybean parameters. And when I run this module, there is an error being shown. And what is this error is? We'll go into the error. They're saying that the module requires QABs, it requires tomato water stress, tea leaf, GBW, RH, and temperature. So that means the soybean parameters doesn't have all of that. So we can check if the soybean parameter has these values, but you will see that soybean and dollar, if you look for temp, anything starting with T, there is only T base, theta is there. The polynomial coefficients for theta, the temperature response function that will give us the function of theta as a function of temperature are available. Theta, the baseline theta, the first order coefficient, second order coefficient. You have TPU rate max, you have leaf transmittance. You don't have any temperature over here. Again, if you see QABs is there, you will see that there is no QABs that is showing up. And if you see any term like stomata water stress, no, there is no term that is, having, yeah, S. If you see S, starting with S, there are a lot of parameters, but you don't have stomata water stress here. So in order to do that, what we will do is, we will, along with the C3 parameters, now we have to give in the input quantities. So we will define the input quantities as, you can see that when I put input underscore quantities, that itself will come. Can you see here? Input underscore quantities is equal to evaluate underscore module. So this is a argument that comes in the evaluate module. This will come like this, and inside within the input quantities, I'm going to create a vector along with soybean parameters, the terms that they have asked me to put here. QABs. QABs is nothing but the absorbed PPFT, which I am going to put it as 1800. Then the stomata water stress. Stomata water stress is the water stress factor that is taking care of the soil water stress. So I'm assuming that there is no soil water stress. And then tea leaf, I'm going to keep the temperature of tea leaf as 39 degrees. 39 degrees, and the GBW, the boundary layer conductance, as around 2.9, and the RH value, that is the relative humidity, as 75%, so 0.75, and the temperature. Now, since the tea leaf is 39, say let's keep the temperature of air as 40 degree. So all these parameters also I included along with the soybean parameters. Now, for example, if you hadn't put these soybean parameters and only the module, let's see what will happen. When you will evaluate the module, you will see argument input quantities is missing. So we'll put input quantities, and suppose we are not putting any other parameter. We are just using these parameters, QABs and everything. When you run it, see, they will say the atmospheric, the carbon dioxide concentration in the atmosphere, GS minimum, G star EA, G star C, J max EA, J max C, KCEA, KCC, all these are required. And all if these are available in the soybeans parameter list. So that is why I don't have to re-enter it anymore. Now, when I do the evaluate module, you'll see that I've got an output, and here is the output of it. You get the total, the assimilation, net assimilation is 26.3676. Conductance is this one. The CI value is this one. CS value is this one. Gross assimilation is this one. Stomatal conductance. The relative humidity on the surface and the photorespiration rate is this one. So this is how the this is where exactly the C3 the temperature response functions are used in Biocro, or basically in the process. It is majorly used in the in the simulation of this AC, AJ, and ATPU values. And C3 assimilation module is the module in Biocro that makes use of these values. Okay. I'll take a break of 2 to 3 minutes. It's 3:48 now. I'll be back by 3:50 or 3:51. I need to have some water, so I'll be back by that time. Okay. So this is how this is the module in BioPro that makes use of the C3 assimilation, makes use of the temperature response functions of Arrhenius equation, John Eyring Williams equation, and the polynomial temperature response curves. Now, what I would like to do is, um, we have the parameters of soybean from BioPro. So I would like to estimate these values of G star. Now we know from these equations that Kc, K0, G star, and Vc max are all of these values. So using all, I would like you to estimate these values by finding out what is the C and EA value. So if I'll just show one example. So if you remember when you're accessing a data frame or a list or anything, you have to make use of the dollar sign. So here, if you want to get the data that is corresponding to uh, G star in some soybean, you can do soybean dollar G star, and you see G star C is available. And if you type enter, you get the value 19.02. So the first step I want you to do is give me all the G star value, the these values, the values of uh, G star C, G star EA, K naught C, K naught EA, KCC, and KCEA. First give me the values, all three values from the software, from the RStudio. Just retrieve extract the values and put it in the chat box. And then I'll say what is the next step that you need to do. By the time you do this itself, you'll be familiar as to how to get values from a data in R. No problem, it's okay. You can ignore that warning. You can just continue working. Are you able to do access something and answer? Let me know if you're stuck somewhere. Okay, no problem. Now are you able to load the library? Is the library getting loaded? Okay. You just have to type soybean parameter this thing and I put it in the chat. So and then after the parameter something like this. Type this, you will get the value of EA and whatever corresponding all of those values. Now you see we what you want is G star EA, G star C, KCEA, KCC, K not EA, K not C. So just get all those values and just put it in the chat box so that I can verify that you're doing it in the correct way. Are you able to do it? Just can you copy paste the error in the chat? Can you copy paste the error into the chat, Ananta? Can you please post the error? Okay. C3 estimate could have find function. C3 estimation model. No, this is the function that you're using, C3SN. That is not the function that needs to be used. You need to use evaluate model. You don't have to, what I asked you to do right now is just get the parameter value. So here in the console, just look at the screen in the console, in the below part, just type in soybean. It's not soya bean, it's soybean. So when you type soy itself, all these options will come. So uh, take this option soybean, enter or click on it, keep a dollar sign, and then you will get parameters. Select the parameters, again dollar sign and G star. That time you will get G star C, G star EA, all of that. Press G star and enter, you will get the value of G star. That is the only thing that you need to do now. Don't run the module and all. Just type in soybean, dollar, parameters, dollar, G star underscore C. You have to be careful about um, the spellings because that is the data that the model will recognize. So I'm copy pasting this thing in the chat. You just paste this in your console and then you will be able to access the value of soybean G star. Now if you want to access the value of EA, you type in EA. Just get me the values. I just need these values. So if you keep on giving these parameters here, you will get all the values. EA. When I'm typing E itself, this parameter is already coming. So you can just enter, enter. So you'll get the value of G star EA. Don't do anything else. Just get me the values. So what values I want is of all these one. The G star EA and G star C I just now told these values KC and K naught C and EA values. Are you able to run it or are you still stuck somewhere? Hi Anamta, have you given up or are you making any progress? Okay, these values I have put in my screen. KC, EA, KC, C, KC, these four values. It's showing null. Okay. Maybe you just copy paste what you have typed this line over here. Can you copy paste and put it here? Okay, it's okay. I'll send in the code. Maybe you can just run those codes and learn from that. So, getting the parameters is just you just need to put in this soybean. It's soybean. So soybean parameters dollar G star. So here I have G star C. If I enter, I get the value. Then again, I can just put all the variables here. G star C is this. Then G star EA is again soybean dollar parameters dollar G star EA. There. G star EA values available. G star C values available. Now I need KC dimensionless parameter, and for that I copy paste this over here and I go KC. Here KCC is there, so I get KCC's value again, I want to get the parameter Kc naught, KCEA, not Kc naught. So I have KCEA over here. Enter, and I get the KCEA value that is shown here. And then I want K naught C. Similarly, K naught EA. So these are the values I need. And all of here I put this, copy paste this. KC K naught C and K naught. So when you press tab, whatever is relating to this K naught will come. K naught E. And I'll run these lines and I get all these parameter values over here. Now, so now we have all the parameters Kc, K naught, G star. Now I also need the ones for Vc max, the Vmax value. So similar to this, I will put in Vmax and something like this, where I get the V values, Vmax 1. So there I got, see the error is coming because I put two arrow marks. Now I want Vmax C. C and it to Vmax or V. V, yeah, Vmax C. Now I want the Vmax EA. And I put here Vmax EA. Again, two arrows, so it has been showing errors. So run. So I got G star's values, temperature response parameters, KC's temperature response parameters, K naught's temperature response, and Vmax's temperature response. Now, using this, what I want to, what I was intending to get done is to use these values to estimate the value of G star. Now we know that G star C is 19.02, G star EA is 3780. So by mathematical calculation, you can find out the values of G star and Vc. All these values you can find out. So let me just here, if you see, you can see the screen. These are the values of G star, C, G star EA, KCC, KCA, K naught C, K naught EA. What I want you to do is find the value of KC, K naught, G star, and VC max. Can you do that, Anamta? This is just calculation. You just need to use a calculator, substitute these values, and find the values of KC, K naught, G star, and VC max. Did you understand what I told? Okay, so just calculate this KC, K naught, G star, and VC max values. All these parameter G star C is basically this C, G star EA is EA. The value of R and TL is this one. Let me get that for you. This is the value of R. R is this value. And TL should be in Kelvin. So you, with whatever temperature is provided here, TL provide 39. TL should be 39 plus 273. Three, one, two. Kelvin. If you get the Kc value, just let me know. Then I can continue. Are you able to do the calculation? Okay, so you can verify with my answers. I'll just show my answers. So here I got Kc as 1684 ppm, K naught as 531.21 concentration of oxygen, G star is 84.45, and Vc max is 352. Now, why I wanted these values is because if you go to Biocro, and you want to, I was talking about how we can use this in the C3 assimilation module. So when we get these values of this one, and if you see the module, if you see what is the output of C3 assimilation module, you can see that assimilation module is giving the net assimilation, the stomatal conductance, carbon dioxide on the surface, relative humidity on the surface, CI value, gross assimilation conductance, and photorespiration. Now, using this, if we need to make an AQ curve, we can create an AQ curve with this module, because this module is allowing for inputs of light and dark respiration and temperature and tea leaf temperature. Now we also have a module called the FPCB module. So theoretically, this module also should allow to generate the uh, AQ curve and AT curve. But unfortunately, if you see here, the module input quantities, you can only vary the CI values. There is no light input, or there is no option for a temperature input. So this FPCB module, at least in this BioCro, this module will be apt for generating the ACI curves. But if you want to generate the AQ curves, then you will have to depend only in C3 assimilation module. So using C3 assimilation module, you can develop AQ or ACI or AT curve. But the advantage of using the FPCB module to generate the ACI curve is you can get all these values of AC, AJ, AP, and all, and see what are the limiting factors when it comes to this. So let's see how this ACI curve varies when we use the FECB module and the C3 assimilation module. So in C3 assimilation module, when we want the ACI curve, we have to define a variable ACI curve, and we are using the C3 assimilation module, C3 assim, and using this function module response curve and giving the module name. We have to give three arguments: the module name, the fixed quantities, and the varying quantities. So in the fixed quantities, I'm giving all the quantities that are corresponding to the soybean parameters. So all the G star, G, KC, KCA, all of these values will come from the soybean parameters. Additionally, it requires a stomata-water stress, the leaf temperature, the boundary layer conductance, relative humidity, temperature. The quantities that is going to vary, light also it requires. Q absolute is equal to 1800. And the parameters that are going to vary are going to be the uh, atmospheric data, CATM. So I'm going to give an atmospheric data of 50 to 2000, a sequence of 50 to 2000 with an interval of 100. That is what this function SCQ will do. SCQ will help you to create a sequence of number that starts from 50 that goes up to 2000 in intervals of 100. So if we create this ACI curves, you get one data frame with data corresponding to all this. Now, when we plot an ACI curve, we need library, the lattice package to make plots. So we will load that library, and we will create a curve, ACI curve, uh, ACI curve plot C3SN module, and XY plot. In the y-axis, we want the value of uh, AN. So we do ACI curve assimilation dollar A assim. And in the x-axis, we want ACI curves CI value. type is equal to L and auto key. Auto key is when you can use the legend. You put it as true, and when you generate this curve. Now, if you want to see the curve in the plot area, you need to print it. So then you will get the plot of the ACI curve over here. So this is how the ACI curve is in terms of the C3 assimilation module. Now let us try doing the same with the FPCB module. Now except here in FPCB module, what is not possible to do is, this is the FPCB module data. I mean the code for running the FPCB module response curve for FPCB module. Fixed quantities are all of these. Now these quantities are going to be helpful because we found it out over here. 168453184 352. Some values we are assuming. So G star value we got, KC we got, KCI, let me put it 1684. Then K naught I got, OI is 210, TPU I'm assuming some value 4.3, VC max is 352, alpha TPU 0, J is 110, RD this value electrons per carboxylation, and I don't have the option to change the atmospheric carbon dioxide over here, because if you see the input quantities, atmospheric carbon dioxide is not an input value for here. So what I do is I will go by the uh, CI values, variation of CI values. And if you see the ACI curve that I've generated using the C3 assimilation module, my CI values, let's see how the CI values are varying. CI values are varying from 40 to around 1600. So what I'll do is, instead of CA values, I will put from 40 to say 2000 by 100, and then I will run this module, FECB module. And now I want to again plot a curve, ACI curve plot using FECB module. And this will change here to become FECB. Here also FECB's output is AN. Then here it will be again FECB and CI. Now if you plot here and print this curve, run, and this is the ACI curve I get for this one. Now I can see how these curves, the results in both of the curves are varying. Now XY plot ACI curve of C3 assimilation module. Assim plus FVCB A, AN, and the y-axis goes from FVCB's CI. You can see that this is the variation between the two curves I used by generated by the assimilation module and the FECB module. Let me put type. Type is equal to L here. So you can see this is the variation between the assimilation module and the FECB module. So FECB module, we use the direct values of G star and all of that. Here, they're using the values of the temperature response function. Additionally, C3 assimilation module can take the values of Jmax, Vmax, etc., and calculate the temperature response also. But FPCB module, directly we have to input one value of TPU limitation and the J value. Now, if you see this equation over here, J is a function of the light, it is a function of theta, it is a function of phi. So all of these are actually it has to be included, but I guess hard coded in this in the FECB module. So FECB module in BioCro can be used just to check how are these curves, what are the values of AC, AN, AC, AJ, and AP, which is the limiting curve over here. So if you plot a line for XY plot of FPCB's AC plus FVCB's AJ plus FVCB's AP plus FVCB's AN versus the FVCB's CI, which I is equal to L, then you will see that there are three curves, and one is and we also need to know what is the photo key is equal to true. So if you zoom this, you can see that oh sorry, what happened? Okay. Here, where is the okay? I will put this auto key here. Auto key true. And if you zoom the curve, you can see that in this curve, you will be able to see that AC is this blue line, AJ is this brown line, and AP is the limiting factor over here. So that is why the AN will be majorly limiting over here. In this portion, AJ is limiting, and in this portion also, I think it is AC that is limiting. So this FPCB module can be exclusively used for the ACI curve, and I don't think so. It is a good tool to use FECB module in BioCro. I'm not saying the equation is wrong, but this module here will not be able to replicate the AQ curve. So the best module for studying all this AQ, ACI, AT curve will be the C3 assimilation module that is able to take in all the temperature response functions also, as well as the weather inputs required for AQ. So this again was an exercise just to help you understand where the temperature response function is used in BioCro. And I think it is necessary, and when it comes to plan modeling, it is necessary in order to have a hands-on experience, you work with the model frequently, because every comma, every bracket, every case, I mean, if it is an uppercase and lowercase, all of that has an impact while when you do coding. So getting familiar with all of that will take practice. So maybe you can use all those sheets that I've sent you, the codes that I've sent you, just to practice by typing it on your own. I mean, copying pasting will again help you get the result, but it will not make you familiar with the I mean, familiar with how to get the I mean there are always situations where you want to cross verify some certain values, where you want to find the what does the meaning of function mean. So in that case, in order to get a practice, it is better you always keep typing and doing the coding rather than copy pasting. Now tools like ChatGPT and all are available. You can just tell them and they'll give you the code. But in order to have a good practice, it is better you try figuring it out the coding process on your own. These are all just very simple commands to get the information from data sheets from the BioCro module itself. So I'll post this in the group, and you can get familiarized with it. And it takes a bit of a time to get adjusted to this coding process, especially if you are not from a background of coding. But don't lose hope. This is very simple. Once you get a practice of it, you will be able to. It is the same code, so it's just a matter of you learning how to familiarizing with all these techniques like dollar sign and all this, to the brackets and the you know double quotes when you use characters and everything to be able to work. So don't lose hope on that. If you're interested in pursuing it, try all these small exercises, and then eventually I think using this biocro module will help you to learn R in a more better way than just sitting and learning R exclusively and then using biocro. So that will be for today's class. Thank you, Anamta, for attending the session. Thank you for trying. And yeah, if you have any doubts, you can post it in the group. And yeah, I think we'll see you in the next class on Monday. Thank you for attending the session.